So the pollen on their legs, you called it the probicular pollen?
Correct, corbicular.
How do they remove that?
So it's actually a very interesting structure. So kind of the mid portion of their hind leg is this kind of like flat dish-like structure, and it has a bit of a divot that actually holds the pollen. And then along the sides, you have these thick hairs that actually come up almost like a Venus flytrap and wrap around the pollen. And that's what holds that nice big granule in place. Pollen is very sticky, so it likes to stick to itself. But when they get back to the hive and they have to remove it, essentially all they do is they just scrape it off from a different direction that isn't the hair. So if the hairs are coming up the sides, you can scrape from the top down or from the bottom up, and it just falls off as one big granule. And that's actually how I remove it in the lab under a microscope, is that I will hold it with forceps and I will use, it's just a little metal scraping device. It's got a wood handle and it comes to a little point, and I just push it off and the granule pops off as one big piece, and then I put it in a tube.
And can you explain again what the process is after you've collected the pollen? Like
So
after you knocked it from the bee?
Right, so after we've taken the pollen, we keep the two different types of pollen separate. So one is washed off of the body, and that liquid is then transferred into what is called an Eppendorf tube. And they're these little autoclavable and centrifuge-ready tubes that we store everything in that are sterile. So one receives the liquid that has all the DNA off the back of the bee, and then one receives the pollen granules. And so once we have all of our pollen, what we'll then do is that we will put it in something called a bead beater, and we use these hard beads and shake it vigorously in this machine to break up all of the pollen cells.
A bead beater or
bead beater?
Okay.
So put in these little beads. I'm not entirely sure what they're made of. I don't know if they're ceramic or something a little bit more dense and hard, but they're just kind of like little tiny beads, and it just shakes it. And that breaks up the pollen, because pollen is actually very hard to get through. It is a single cell, and it is just tough, that cell membrane. But once we beat it up, then that's what allows us to actually get to the DNA, because we have to get through that cell membrane. And so we have an extraction kit that goes through several processes where you're essentially using enzymes to dissolve what you don't want and keep what you do. And of course we want the DNA, and then you wash it, and at the end you're left with really just your DNA. And then you put it through something called PCR, which stands for polymerase chain reaction. And it is a way in the lab to replicate DNA like it does in your body, using the enzyme DNA polymerase. And then you supply that with all of your nucleotides and your primers, because DNA happens both in forward and reverse, so you need forward and reverse primers. And then once you add that all, you put it in something called a thermocycler, and that's, since we don't have all of the enzymes that actually replicate DNA in your body, because there's a lot of different players that come together to ensure that this happens in your body. We have to artificially denature the DNA with heat, and so that's what the thermocycler does best. Is it's very accurate in these tiny little PCR tubes, and it will heat it up to denature to allow DNA polymerase to bind, as well as the primers. Replication happens, and then it cools it so that it comes back together, and now you have two. And then it will do this anywhere between 30 and 40 times over the course of about 1 to 2 hours. And so at the end, you have billions and billions of replications of this DNA. So even though we've only collected a small amount of pollen, and in that pollen you only get about a small amount of DNA because you always have lots, we're able to amplify that to large amounts so that we can then analyze it. And then that also ensures that we have a bunch of pollen, so if things go bad, sometimes the PCRs don't work, this actually happened to me a lot in the lab. Sometimes the thermocycler battery's gone bad, so it stops halfway through and you don't get proper replication, or the dye that you're using in your gel is bad, and so you don't actually get to see your DNA under the UV light. So a lot of things can happen, but by replicating it a lot, you don't lose it, right? Because it's difficult to come out here and sample all of these bees, and it's not something you can just do again. And then we we sequence it. And so that's what we're going to a collaborator for at a different university. And the sequencing is essentially a PCR, but all the nucleotides are labeled with a specific colored fluorescent dye that will give off a specific color when shined by light. And so since all these nucleotides are labeled, DNA polymerase still arranges them in the proper order for the actual loci, which is the DNA. But now that they're labeled and we shine it through a light, each base pair will give off a certain light, and the machine can read it and tell us exactly what each base pair is in this loci. And so then you can take that and compare it to other literature, and that's how we determine what plants the pollen came from, is by comparing that segment of DNA in that specific order to what we have on other plants already collected.
Awesome.